It's 1 o'clock, so we're gonna get started again here. If you folks could make their way back into the room, and we've got another group of speakers this afternoon, so we're gonna be hearing about some different things that related to insects, disease and modeling again. So we'll be hearing from some different folks, but I wanted to revisit kind of what we left off with at the last session about kind of the thought of what agencies are doing, what's the opportunities, and we had a really good exchange there at the end with NSF and some of the opportunities. And I'm hoping that we can hear from NIFA. I know Nancy Cavallero was here, and I don't see her right now, but we'll possibly, when she gets in the room, maybe we can cut to that, because I know she has to leave again this afternoon. So let's not delay this, though. So I'll just turn it over to Jeff Hicke from University of Idaho. And he's going to talk about some of his work on forestry modeling and with insect pests. So Jeff, whenever you're ready, go ahead.
Ready to go. So thanks very much for the invitation to speak, Steve. Enjoying the conference. Unlike many of the other talks, I'm not going to give an overview or a broad review. Instead, I'm going to focus pretty specifically on a case study that we've been working on for several years in my lab. Although at the end, I'll try and take a few steps back and generalize some ideas that we've had. Matt yesterday talked about a clear example of climate change impacts on forests in the East Coast with the example of southern pine beetle. I'm going to talk about what's probably the best example of climate change impacts on western forests, and that is the the upward expansion of mountain pine beetle into whitebark pines in the western U.S., and in specific in the Greater Yellowstone Ecosystem. So I'd like to acknowledge my co-authors. Polly Biot was a PhD student of mine whose work this talk is about. Hagenish Preisler, Ken Raffa, Jesse Logan, and John Abatsoglu have contributed significantly as well. And then funding for this has come from the Northwest Climate Science Center and USDA NIFA through the Forest Mortality Economics and Climate Project. So starting broad, beetle outbreaks are significant forest disturbances in western North America. The map shows you locations where beetle outbreaks have occurred in the last few decades. The different colors show different beetle species and tree species. Billions of trees have been killed according to our estimates, and they represent about 7% of the forested area in the western US. You can see from the map, although it might be hard to see, that most of the forests of the western US and of BC, for that matter, have been affected by some level of beetle activity, with the gray areas showing places where they haven't been attacked, and that's been mostly in the coastal areas of Oregon and Washington. Beetles have killed more trees, we think, than wildfires in this time period, and this amount of mortality represents about half that of harvest. So pretty substantial amounts of mortality. I'm going to focus on mountain pine beetle, which is green on this map. I'll come back to this map. Mountain pine beetle is fairly well known in terms of its drivers, including the influences of climate. And I thought I'd start by going over some of the factors that are related to mountain pine beetle epidemics. We've heard about bits and pieces of these in different talks, but I'll just be a little explicit. There's a set of factors related to the trees themselves. The beetle species prefer or are obligate on certain tree species, although mountain pine beetle attacks a number of different host pines. So there needs to be a presence of host tree within a stand. And the next set of factors, the next three factors, are related to a tree's capacity to defend itself against attack. So stands that are crowded and therefore there's a lot of competition among trees, older trees, larger trees, and then drought stress on trees all reduce trees' defensive capacity. So climate, you can see there, operates on trees through drought stress. There's also a set of factors related to beetles. The beetle populations last year are important, as Matt mentioned, whether that's locally or in a dispersal sense from other stands. And then the second set of factors that or influences of climate are related to direct temperature effects on the beetles themselves. And those come in two different processes. One is that very cold conditions in wintertime kill beetles that are under the tree's bark. So that's the first, winter beetle mortality. The second is associated with year-round temperatures that switch beetles from a 2-year life cycle to a 1-year life cycle, as well as synchronized populations. And that's important because these beetles need to mass attack trees in order to overwhelm tree defenses. And it's thought that the temperature year-round actually synchronizes these the populations so that they can mass attack. So this is called adaptive seasonality. So you can see the three factors up there: drought stress on trees, winter mortality, and year-round temperatures. I'm going to focus on the role of mountain pine beetle outbreaks in one particular tree species, whitebark pine. Yes, Jeff?
I'm sorry, can you just say what do you mean by year-round temperatures?
Oh, sorry, yes. So the question is, what do I mean by year-round temperatures? That just means that temperatures in every day of the year, effectively. So not just in summer, not just in winter, but the temperatures influence the life stage development of the beetles, and that occurs throughout the year for this particular species. So I'm going to focus on whitebark pines, which there's not a lot of them throughout the western U.S., but they are ecologically important in a few ways. They exist at the highest elevation for tree species in the U.S. Rockies, particularly the northern U.S. Rockies, as well as the Cascades and the Sierras, and their range actually extends into British Columbia. So they're important because, as the lower left figure shows, they act as a nurse tree for other species that otherwise might not be able to establish and withstand the harsh conditions at the high elevations, which are windy and cold. So that's one role. And then the other figures show that the whitebark pine is an important food resource. So on the upper left is the Collick's nutcracker, which is a bird that has a symbiotic relationship with whitebark pines. Squirrels also harvest the cones and collect these cones into large middens that can be several feet across, 6 to 8 feet across. And those middens are then harvested by grizzly bears, or raided, I guess is the way to say that. Raided by grizzly bears at a time when grizzly bears are building fat reserves for their hibernation. So there are interesting ecological implications for whitebark pine and its mortality. Whitebark pine has been recommended for listing as threatened or endangered by the Fish and Wildlife Service as a result of three different threats. One is that the climate change, or expected climate change, is going to reduce the suitability for habitat for beetles, for whitebark pines in the future. There's also an invasive pathogen that causes white pine blister rust that's killed lots of whitebark pines as well as other pine species in the West. Fire suppression has led to the growth and overtopping of whitebark pines by these slower growing tree species, particularly subalpine fir. And then the last one, which I'll talk about, is mountain pine beetles, which you can see from the photo there, which is in the Greater Yellowstone area, have killed substantial amounts of whitebark pine. Okay, so what's been going on recently? The map shows you in red the locations of whitebark pine mortality from beetles, mountain pine beetle. You can see there's a lot of it spread throughout the western U.S. The time series just is an indicator that over 1 million acres have been affected by these beetles. And we know, these are maps from 1997 forward. We know that outbreaks have happened in whitebark pine in the past, in the 1930s in particular. There's a good example. But we also think that the extent of the outbreaks in the western U.S. has been more severe in the recent outbreak, more severe and extensive. So we think something is going on that's unusual, but it's not entirely naive. That is, the host isn't entirely naive. So I'm going to focus on some work we've done, in particular in the Greater Yellowstone Ecosystem, which is the red box there. Jesse Logan has done a lot of work actually on mountain pine beetles and whitebark pine and other pine species. The figure on the right is a time series that shows in the blue line the winter minimum temperatures, so that's associated with beetle mortality, back from 1920 to the present, and a process model that predicts survivability based on these temperatures in red. And so you can see there's this shift in 1980 that suggests that conditions became much more favorable for beetle outbreaks. On the left is the year-round or mean annual temperatures, also indicating that in about 1980 or 70 or 80 that conditions warmed substantially in the area. So we built on Jesse's work here, which Jesse's work that I present is a look at these individual climate factors. We wanted to take a look at what the integrated picture was. I mentioned all those factors in a previous slide. And we wanted to let the data, that is observations of beetle outbreaks and climate, tell us what was happening. So we performed a statistical or empirical analysis, and I'll describe that now. Whoops, not now, but after the study questions. So we were interested in three main questions. One is, what were the climate-beetle relationships? These are nonlinear relationships, as you'll see, so it's not just as simple as a linear relationship. We were interested in what the functional forms were. The second, we were interested in the recent outbreak and trying to identify which of the climate factors were most important in facilitating that outbreak. And then third, we used projections of future climate to estimate what would happen in the future in this area. So to build the statistical model, we used observations of tree mortality from mountain pine beetle, from Forest Service conducted aerial surveys. You could see the map there. These are, we gridded at 1 kilometer spatial resolution, and we had several decades worth of data. So that forms the response, or the independent variable, the presence of whitebark beetle mortality in whitebark pine. And we also used this to look at what were the beetle populations last year, which is an explanatory variable. On the right side, you can see we used a variety of climate data sets, the observationally based gridded data set of PRISM, as well as BioSim, which is an interpolation scheme looking at daily data. And BioSim also allows us to run, or allowed us to run, process models, one of which I showed you earlier that predicts suitability or survivability of winter conditions. And then we also used, where possible, maps of biomass or age or size, or the amount of whitebark pine within a stand, or the remaining whitebark pine after mortality happened. I will say these are not as extensive, or we don't have as confident in the information in these stand structure data sets, and so I think that is a bit of a weakness in our study. Okay, so we developed a model. And the first thing we want to do is assess how confident we can be in the results. So this is the time series of area with mortality on the y-axis from 85 forward. And the black dots are the observations. And the red dots are the model predictions from a cross-validation exercise, with the dashed lines indicating the confidence intervals. So we feel that the model is doing a pretty good job at representing the observations, and therefore we have a decent amount of confidence in what the model is telling us. So to answer the first set of the first question about the climate-beetle relationships, I'll show you a series of plots that look like this. On the x-axis is the explanatory variable, in this case winter minimum temperatures. The top plot shows the probability of tree mortality from beetle outbreaks. And the bottom plot just shows you the frequency distribution of the observations that went into building the model. So as expected here, the winter minimum temperatures, as they increase from -25 to about -15 degrees C, probability of tree mortality or outbreaks increases substantially. But then after about -13, -12 degrees C, the line is pretty flat, indicating that there's no response or no influence of this variable on outbreaks, which makes sense because this is a freezing mechanism. And so once the temperature gets above a certain amount, any additional warming really has no effect on whether beetles survive a winter. Okay, the second important variable is drought stress in trees. This is, in our case, the best variable, was represented by 2 years of summer precip, so last year and this year. And you can see as drought stress increases, that is, as precipitation decreases, the probability of tree mortality goes up. Again, not surprising, but it's nice to know that the data are telling us this. And then year-round temperatures, we actually divided into fall temperatures versus spring, summer to try and parse out those effects. So fall temperatures on the left, spring, summers on the right. And like winter mortality, fall temperatures seem to have an influence at low temperatures, so below 0 degrees C on average. There's a sharp increase in outbreak probability when temperatures increase, but above that, there doesn't seem to be much of an effect. And in contrast to the previous three examples, climate variables, this spring-summer temperature really didn't seem to play a major role. So either the confidence intervals were really wide, as you can see at 0 and 2 degrees C, and or the line is fairly flat, indicating no response of outbreaks to this variable. So I'm not going to talk about this variable anymore. So the second question we were interested in is which of these factors were influencing and at what times were they important during the recent outbreak? So the recent outbreak, well, is defined here. This is a time series back to 78 through 2010, and the dashed line indicates the area of the outbreak. So you can see it's really ramping up in the early 2000s, peaking in 2008. What's plotted here is what we call a climate suitability index. So from that statistical model, we extracted just the climate variables and added them all together to look at how suitable climate was as a whole for mountain pine beetle outbreaks. You can see that early in the record, about the first half, there were multiple years when the probability or the suitability index was quite low. It didn't happen every year, but it happened frequently enough that we think that kept populations in check. But then starting in the late 90s, early 2000s, you can see that the climate suitability index is fairly high and consistently high for multiple years in a row, leading to at least facilitating the outbreak. So we can unpack or break down which of these variables were important at which times. And the first one I'll show you here is the minimum temperatures in wintertime that influence beetle mortality. So early on, you can see that there were multiple years when suitability was low for outbreaks. These are years when there are very cold conditions. Cold snaps have occurred or did occur, again, keeping beetle populations in check. But since 1995, the Greater Yellowstone Ecosystem experienced very warm winters only. And so that allowed increased survival of beetle larvae under the bark. We think that was probably one of the most important reasons. We also think summer precipitation, the role of drought stress, did play a role. You can see there's no directional long-term trend, but it did go up and down. We had droughts in the mid-90s, and especially in the early 2000s, there was a west-wide severe drought that led to high suitability for beetle outbreaks. So in this case, high suitability means droughty conditions. And you can see that drought occurred early on in the outbreak and allowed lots of trees to become susceptible to attack. And then finally, the fall temperatures, which was the last of those four variables that we thought were important, seemed to be continuously suitable. You can see the index was fairly high throughout the record, maybe a couple years in the mid-1980s that were low, but generally we don't think this was a limiting factor, at least back to 1978, aside from a few years. Okay, so now we have the model. We looked at the recent outbreak. What happens when we apply climate change projections? So what will we expect happen in the future? Again, we're focused on the suitability index. That's a index of how favorable climate is. So I'll show you a couple different time series. These are time series that go back to 1950 forward to 2100. The gray and black line, that's from 1950 to 2010, is the PRISM data set to give you an indicator of what the observations tell us. The dots are the multi-model means, so we used 10 different models that we selected based on their fidelity, their ability to replicate historical conditions. So there we have a multi-model mean, and this is a RCP 8.5 emissions scenario. And then the gray envelope shows you the range of those 10 different models. So not surprisingly, with winter temperature, warming in the future is projected. You can see the dots move to continuously suitable for many years in a row. The gray envelope tends to shrink in terms of removing lots of those years that have low suitability. Precipitation is a different story. The multi-model mean, again, has no long-term projected trend to it. And so what happens is the suitability really depends on the year and the climate model. So the climate model variability is illustrated by these two different lines. One in red is a model that's got a drier than average projection for the future. And you can see this red model has higher suitability throughout the projected time period. In contrast, the blue model is typically the model, or often the model, that has the lowest suitability in terms of precipitation. It's the wettest model. And so you can see there's quite a difference in how models project what will happen in the future with respect to precipitation and therefore the effects of drought stress. And then summing these two together, the integrated effect is driven primarily by this increase in suitability due to winter warming, with the variability in precipitation added on top of that. So it doesn't look very promising to be a whitebark pine in the Greater Yellowstone ecosystem in the coming decades. Okay, we can also project what will happen spatially. So our models run again at 1 kilometer spatial resolution. What's shown in the upper right, what's shown in all of these in terms of colors is the fraction of years within a particular period that are suitable for mountain pine beetle outbreaks. And we're just focused here on winter temperature, given the directional trend. And so in the observational period, upper left, we see that there's quite a bit of spatial variability. The blues indicate locations that have low suitability. The reds indicate locations that have high suitability based on this particular climate metric. And you can see there's quite a bit of blue, especially in the southeast, which is the Wind River Range, a pretty cold environment compared to the rest of the Greater Yellowstone ecosystem. But you can also see as the years progress that the conditions warm up in general, and that the blue colors eventually shift to red colors, such that most years in most locations tend to be suitable in terms of this winter effect on beetle mortality. So what I've shown you is information, a lot of information about our study that really isn't new. Lots of these drivers and influences have been documented in the past. I think what's different about our study is that we looked at a broad region here, and we allowed the data through a statistical analysis to tell us what the relationships were and what the importance was in an integrated fashion. So we considered all the major drivers. Okay, so a little bit about what's next for us. We were focused on the Greater Yellowstone ecosystem. You can see from the map that's in the lower right side. Whitebark pine occurs in other locations, as illustrated here. And we know that as one progresses from the interior of the continent towards the coast, the Cascades, that for instance, different climate variables change. So in this case, I've just indicated that the winter minimum temperature, this important variable, shifts from minus 16 degrees C on average in the Greater Yellowstone ecosystem to minus 9 degrees on the Cascades. So that suggests that there's possible variability in terms of which drivers were important in influencing the outbreaks that we know happened in this region and we expect might happen in the future. And this has been noted by Aaron Weed and Matt Ayers and Barb Bentz in their paper recently, where they just indicated that some regions did have an influence in terms of this winter minimum temperature, and other regions it was just consistently too warm. So we're interested in assessing that variable as well as other variables. So that's a bit of a broader spatial context. Also interested in another host species that's important economically and in other ways. This is lodgepole pine. So again, mountain pine beetle, now lodgepole pine, and that's all the green areas on this map. Lodgepole pine, unlike whitebark pine, is a harvestable or a timber yielding species, so it's important economically. It's important for carbon and nitrogen cycling and water cycling because the extent of tree mortality is so great. You can see in British Columbia, huge areas were affected. So there's a ecosystem effect just based on the number of trees, and there's a human impact because of especially recreation values and timber values. Lodgepole pine is different in the sense that it's evolved over millennia with mountain pine beetle, unlike whitebark pine, which is maybe only sporadically seen outbreaks, if at all. And so it's a less naive host that might be better defended. And so there might be some interesting differences in relationships compared to whitebark pine. And then you can see there's a suite of other bark beetles in the west that have attacked and killed lots of trees: Douglas fir beetle, spruce beetle, western balsam bark beetle, Ips engraver beetles, and so on. We expect that the climate effects are likely to be similar. Temperature plays a role in influencing life stage development rates, ability for a beetle to survive winters. But we also expect differences among these as well. So emergence is perhaps at a different time. There might be multiple life cycles per year. The role of drought is stronger in some beetle systems than in other beetle systems. So we're interested in trying to assess how generalizable might the climate influences be on multiple bark beetle systems. Okay, so to summarize, we looked at specifically mountain pine beetle outbreaks in whitebark pine in the Greater Yellowstone Ecosystem. We confirmed the previous studies that indicated that warming winters played a major role in driving these outbreaks, and that drought, especially early in the 2000s, kicked off the outbreak by making trees more susceptible to attack. And this is a clear change of climate change impacts, at least on the temperature side. Warming throughout the western U.S. has been attributed to anthropogenic climate change. Drought, in particular this drought, to my knowledge, has not been attributed to climate change, except perhaps through the temperature effects, but the temperature is. So this is pretty visible to folks visiting Yellowstone National Park, for instance. I mentioned bad time to be a whitebark pine, increased or continued suitability in the future. And in related systems, whether they be related in terms of whitebark pines in different locations, or same beetle, different host systems, or bark beetles in general, we expect similar outcomes, but there probably are nuances or maybe even significant differences among the different systems that we need to explore more. So thank you very much for your attention.
Great, thanks, Jeff. Any questions that folks have? Yeah, Ben.
Hi, Ben Cook. So Park Williams, a colleague of mine, has done a lot of work in the Southwest, and he finds the strongest predictor of kind of moisture stress on trees to be actually vapor pressure deficit, which kind of integrates everything happening in the atmosphere and the surface and includes both changes in supply and demand. Have you looked at vapor pressure deficit in your model to see if that pops out more than precipitation?
Right. So we did a variable selection exercise where we represented each of these climate influences with multiple different drivers and then allowed through AIC the process told us which variable was most important. And so we considered for drought stress, climatic water deficit, VPD, year-round precipitation, various lags in summer precipitation. And it turns out that summer precip was the main one.
Steve Frank. So in one of your last figures, there was a 4-panel figure with different colors, and the bottom right one was 2070, this one, to 2099. And so you get, obviously, the whole area is suitable or highly suitable for beetle outbreaks at that point. But by that point, have you exceeded the thermal tolerance of the trees? Are there any whitebark pines that exist under those conditions?
That's a good question. We haven't actually looked at the trees and how they would evolve in the coming century in terms of range or distribution. So
it sounds like they're pretty sensitive from your introduction.
Yeah,
I just wondered
yeah,
if
they sit at the highest elevation, so it's likely they're going to move up and fill in some of the, you can see some of the bathtub rings are going to get filled in by whitebark pine, but the beetles actually move quicker than, because of their life cycle, move quicker than but then there's this issue, okay, beetles, trees aren't going to become susceptible to beetles for 30, 40, 50 years until they become big enough. So it's an interesting dynamic about how that will all play out. I will say that some of the upper elevation areas have very little soil. So at some point, they're looking to move up into rock, and that's hard to establish if you're a tree. But yeah, that's a good point that we didn't take into account the movement of whitebark.
Nancy, go ahead.
So I had a question, but I also am going to have to leave pretty soon, so I wanted to get questions from you for me that apparently you had before I got here. But so these different species you were talking about, lodgepole pine, et cetera, it's a different species of bark beetle, or is it just a different variety? Or, I mean, how specific are these bark beetles to the species of tree?
Oh, it's worse
and
than
the
that.
conditions and all those sort of subtleties of where they're suitable, just to try and get this all in perspective here.
Right. I didn't mention that one of the reasons we want to actually do this study, look at whitebark pines outside, is because Barbara Bentz has shown that there's differences among mountain pine beetle populations in their sensitivity to temperature.
Right.
So this is her work is within lodgepole pine. But there's no reason to think that there isn't that population variability in whitebark pine. Whether there's a host, there's a lot of mixed lodgepole whitebark pine stands in the Middle Rockies. My understanding is that we're not quite sure whether beetles are specific to the host that they developed in. So beetles that developed in the whitebark pines, do they actually attack whitebark pines, or do they switch? Can they switch? That is outstanding, but there is that idea out there that there might be some host selection.
Yeah, okay. And then just before I leave, I gathered there was some conversation that I probably should have been here for before lunch, and if we can just take a little break and address those. One of the things that my program at least partially funded this, and one of the things we said in the call for applications for this program was that we're hoping that these conferences will help inform future requests for applications. So as your discussions move along in that direction, where are the needs for further research in this area? So, but I know there were some questions that maybe I could answer.
Yes, I would encourage people to talk to Nancy, because I think that's a very important part of this. And we did have a conversation here with the NSF. So just to remind folks that to interact with Nancy as you can. Were there some other questions specific to Jeff's talk? Andy, did you have a question?
Yeah, so I had a question, and you maybe kind of partially answered it, but so as you see these huge outbreaks and a lot of tree mortality, what happens to the beetle populations, number one, and then what sort of impacts do you see around forest succession, and what do you see around just wildlife habitat in general? Because you kind of pointed that out at the beginning as a key part of the white mountain pine, or the white pine.
Right, so let's see. The first, you asked about impacts to forests. Beetle outbreaks, they kill a lot of trees, but they are unlike a severe wildfire in the sense that they leave behind the smaller diameter trees and not host species. So in fact, forests recover much faster from beetle outbreaks than from fires, and because there's typically a component of the forest that remains and can take over, establish. So succession is not reset to zero, it's kind of set a little bit backwards. Yeah. And I forgot the first part of your question.
Populations of beetles in general.
Right. So what you're asking about crashes, population crashes. So there's a couple of ways that can happen. One is if there's a very cold winter snap. That can knock back populations, and Matt talked about that with southern pine beetle, and it's been demonstrated for a few cases for mountain pine beetle. More often, they tend to run out of food. That there is many of the hosts are killed, basically, and the populations just crash because they can't find enough food resources.
Great. Any other questions? Chonggang, you had one?
Jeff, it's great. I like the work. I just am wondering, like, you consider the temperature, precipitation. I'm just wondering, like, one thing, like, it's a huge component is the CO2 fertilization in the future. So I just want to hear what's your opinion on that, how this type of model can account for that in the future research.
Yeah, it's definitely not accounting for. This is a statistical model. There was no interaction or influence of CO2 that we used. So the role CO2 would play is would whitebark pines or pines in general be less drought stressed because of higher CO2? I don't think we know that. I certainly don't know that.
So I'm just going to ask a question related to, I think the next, you showed the map where you've got that one right there. So, and maybe you already addressed this, but the really dark green is up in Canada. And we've talked about this previously about, what is occurring in different countries. So Canada's doing something, US is doing something, and pardon my, if you already addressed that, but is there related activity up there, or you're obviously working with them, because that seems to be where the area is most intense to this, related to this issue.
Right. Well, it's certainly intense. It turns out that the area of beetle-caused tree mortality is about the same in the western U.S. as in British Columbia, despite the huge blob that you can see there. Are you asking about the causes and humans' influence of the causes or response?
No,
or?
not so much, but what are they doing? Or is it a they, or is your group partnering with them, to address this, regional issue that goes across borders, so to speak?
Well, there is a lot of common research in terms of understanding what the drivers are. There's been quite a number of studies that have looked at the mountain pine beetle expansion, as you can see, north into northern BC, but and was mentioned earlier, across into the boreal forest. So the Canadians are quite interested in understanding what the drivers are, how long climate will be suitable, or will it continue to be suitable into the boreal forest, which is Alberta and to the east. And so we have a lot of common research and in terms of both the climate aspect as well as what to do about it, thinning studies versus not, try and get ahead of the beetles. I'm not sure if I'm answering your question.
Yeah, so you are working with them, there's connections established.
Right, we go to the same meetings, we have collaborations, yeah.
Okay, good. Great, well, that's all the time we have, and we're gonna move on. Thank you, Jeff, again for your presentation. We're gonna now hear from Karen Garrett from the University of Florida, and hear about some of the work that she's been doing. And she's going to actually present something that maybe needs some feedback, so we'll be interested to hear.
Yeah, thanks for the opportunity to present to the group today. It's luxurious to present after so many other people have really laid the stage and explained a lot of concepts and gotten into those. So I'll go over some points quickly where I think other people have already touched on them, and I'll give a little bit of an overview of issues for plant disease and pest management. So I'll start by just some general challenges for how we've been thinking about adapting disease and pest management. I'll talk about one of the focuses of my work, impact network analysis, designing a program to integrate across socioeconomic networks and biophysical networks. And then finally, this point, we've been working on for CGIR, a scoping study for strategies to help resource-poor farmers adapt to climate change. So that part would be interesting to get your feedback on. And maybe it would be have some of the same perspectives as for thinking about the United States as well. So one underlying question that I'll have in the back of my mind that's also interesting to think about is maybe for this group is what constitutes a healthy community of scientists and technologists to support climate change adaptation. Like people have been talking with some about how like, oh, well, it's hard to get funding, it's hard to have people to work on problems. Well, I put healthy in quotes because it's a little bit tricky to talk about what is a healthy plant. It gets harder to say what is healthy soil. That you can debate on that a lot. But it's interesting, maybe harder to think about what's a healthy community of scientists and technologists, but maybe that's part of what we need to grapple with also. And I'll just throw this up too. Sometimes I leave out yield loss and just start talking about plant disease as a problem. But maybe for some members of the audience, it's important to just think about these kinds of losses. So here's one estimate, and well, in a sidebar, it would be that we really lack a lot of good estimates for yield loss. And despite its importance, it's kind of hard to get good estimates for yield loss, and it's not necessarily scientifically exciting topic. What is the yield loss? But it's key to making priorities, right? So here's an older 2006 estimate for major crop plants, and it differentiates between what's the potential loss and then the actual loss, since people are trying to manage, right? One thing that's important to keep in mind, though, that these are mean losses, and for crop species where there have been relatively a lot of research put into it. But of course, the major effects on food security occur when there are the particularly high losses. Like if you have a lot of years where you're getting 1 or 2% yield loss, then that adds up. But the big effects on food security are the major epidemic years. So just thinking in general about challenges for adapting disease and pest management. Some open questions. Also, these are ones that might be fun to discuss some more tomorrow. Because often we think about, well, how can we sustain a system at least as good as ours in the face of climate change? But of course, our current system isn't necessarily optimized in any way. Question some other people were talking about too, is adapting disease management to climate change the same as adapting to climate variability, or one of those different? And I think it's interesting to discuss that. Can you look at no regrets options, the things that even if climate change doesn't follow the trajectory it might, you're still glad you made those investments? And probably for crop disease management, there are a lot of those, right? They're just basic things we'd want to do whether or not there was climate change anyway, and to invest in those. And then a question of how effective can individual decision making be, and when do you need policy changes to really have things be effective at a larger scale? Okay, so thinking about what do we need to understand to adapt to climate change from the context of pest and disease management. So for growers, so they're having to adapt to different circumstances all the time, right? So if they have formal early warning systems or casual, intuitive early warning systems, they'd have to think about how to adjust those within season, and then also longer term as their longer term decision making. So one thing that people would always like would be, growers would always like would be good predictions for how they would adjust, like what variety will they should they choose for this year, to have like those kind of longer term weather forecasts that would allow them to say, oh, well, I need this variety because of that weather prediction. For plant breeders, then a big thing is what diseases to prioritize where. So to what extent can our evaluation of climate change and how that will affect disease risk influence what breeders prioritize? And it's similar for policymakers and donors. What should they prioritize? What should they invest in if it takes a decade or more to get a new variety together? And outside the realm of biology, what kind of financial tools, like insurance, could be used to buffer farmers from increased variability? And then, of course, the natural system, there are a lot of factors that need to be understood, including distribution of resistance genes, which is often a big undertaking to understand. So this was an overview we published a while ago, just thinking about the idea of genomes to ecosystems, what will be the effects of climate change, and just kind of summarizing what plant pathologists can bring to the table for thinking about this. So at the field level, as of course many of you already know, so the key effects of climate on plant disease would be, for example, for fungi and bacteria that infect foliage, temperature and precipitation or humidity are major factors in infection risk. So often, even if the temperature becomes more conducive to plant disease, if there's not enough humidity, then there might actually be less disease. Effects on overwintering and oversummering, the extremes that might allow diseases to really take off. And phenological issues, for example, fusarium head blight, we have to have the timing of the availability of the propagules of the pathogen at the right time in flowering for the plant. So plant pathologists have done a lot of work to understand these smaller scale processes. Then of course there's a scaling problem. So even if you understand what's going on in the plant under a range of different temperatures, say, and its interaction with the pathogen, then you have to try to understand how to scale this up to produce predictions at a larger scale, and then start to include the factors of which species invade. Let's see, so thinking about what you can do within field management, so host resistance, of course, is the best option if it's available, and that goes back to the issue of crop breeders having a decade or more idea of what priorities will be in the future so that they could incorporate those ideas in their breeding programs. Cultural practices can be adjusted, like timing of planting, pesticides, and biocontrol, which is a whole other layer if you want to think about how climate change will affect not only host and pathogen, but then also biocontrol agents. So some types of management can be subject to changes under different climate change scenarios. Like this was a study of using potato mixtures with different disease resistance genes across a climatic gradient. And so this having mixtures, which is a traditional cropping system anyway, has some modest benefits for late blight management, but the benefits are lower in areas with longer growing seasons. So this gets back to the question of the generation time for pathogens, right? So if you have a longer growing season, that can add some additional generations to the reproduction of the pathogen, and can potentially have a big impact on how important the epidemics are. And so for things like mixtures, and then variety mixtures, and then some other methods, like some, depending on the type of resistance gene being deployed, sanitation and so on, those all may become less useful as the season becomes longer. So that's another sort of interaction. It's just the effects of climate change on the system, and thus how viable different types of management are. And this is an illustration for potato late blight of this idea of winners and losers and the effects of climate change. So here in this work of Adam Sparks and our team, we can find, for example, in Rwanda and Ethiopia, some locations where risk for potato late blight would be predicted to increase, but then there are a lot of places where it would be predicted to decrease, again, often because of the reduced predictions for reduced moisture availability. So at the same time that we can produce these kind of maps, of course, we have a limited amount of confidence in them because of all the different sources of variability. But in sweeping terms, they can be used to give some kind of recommendations to breeders, just as they're starting to think about where their program would be heading over decades. And probably a lot of you know this paper. I think it's interesting. It makes the point, crucial component of the approach is the implementation of adaptation assessment frameworks that are relevant, robust, and easily operated by all stakeholders, practitioners, policymakers, and scientists. So the idea of adapting science as well to address the problem of climate change. So we were getting at this, just starting to get at this issue some, where we're looking at an analysis of climate variability and the color of weather time series at a degree of autocorrelation for agricultural diseases and pests. And then under these same modeling scenarios, we were looking at how the different kind of weather scenarios would affect decision making. And in this case, we just had a really simple decision-making model where farmers are either thinking really a lot about what happened last year or thinking longer back into time when they're deciding what to do, which corresponds to some models of human decision-making that people do have a tendency to weight very heavily what happened just most recently. And so we started looking at that sort of idea here too. And it's interesting. Well, the results are complicated, hard to summarize in a short time. But so that also inspired us then to start looking at these kinds of scenarios when you've got a decision support tool. So we haven't published this yet, but we're working to characterize the different types of scenarios where this would be true. because you could have a scenario where, as you go across time, you've got a decision support system that is useful and it continues to be useful under climate change. But then you could have a scenario where you actually need to construct a new decision support tool, or that it's not possible to construct a new one. Well, you could even have scenarios where the old decision support system was not useful, but it becomes useful under the new environmental conditions. So this seems interesting to me to think about what sorts of information we provide for decision making and how it could be modified. Okay, so impact network analysis then, give you introduction to that, and then this scoping study that could be interesting to discuss. So this is a good survey of integrated pest management adoption in developing countries and what the limitations are to it. And it's interesting to see that when people report why management schemes aren't implemented, that often it's just lack of awareness that they even exist. So in addition to other kinds of weaknesses that can be in place. So it could summarize that a common problem is that even if you have a good disease management concept or technology, do you understand how it performs across a region? Will enough people learn about it and choose to adopt it that it'll be effective in regional epidemic effects? Because everybody's decision you can think of as producing a management landscape, and so will that be enough to slow epidemics to provide protection regionally? And then, of course, if you throw in climate change or a new pathogen strain, then it can be useful to think about how the system will have to be modified. So that's the idea of impact network analysis. So our lab's working on that as a platform for evaluating system management strategies, looking at the impact of a research product like a new variety, disease resistance, or even just an idea like change the planting date, and how that affects spatial ecological processes, thinking about regional processes, and how that impact occurs through communication and decision-making networks and their link to the biophysical networks. So if you're not used to thinking about network models in a more formal sense, I'll give you a very brief introduction here. So the idea that you've got a set of nodes and the edges or links that link them together. And so the nodes could be lots of different factors, and then the edges correspondingly. For example, nodes might be farmers, and then the links could indicate communication between farmers or exchange of payment between farmers for purchases. And then also they can change over time. So of course, if you've got some issue like climate change, a factor like climate change, it can be having an effect too. So you can think of a network like this in terms of a social network where people are exchanging information and influencing each other. You can also think of a network linked to that of farms and, say, the spread of pests or diseases among farms. Once you know something about the network structure, you can start to draw some conclusions just based on that structure. Like if you're looking at a network, so this is very simple, it's maybe more meaningful if you have a larger network, but you could look at things like the degree centrality, so the number of links a node has. So these nodes are more important from that standpoint, higher degree centrality. Closeness centrality, how easy is it to get to other nodes from a particular node? Betweenness centrality, so does a node act as a bridge? Like here in the middle, these are bridge-like. How important are the neighbors? So of course, if you have a communication network where you want people to exchange information about management strategies, then you want more connectedness. But of course, if you have an epidemic or an invasion spreading, then you want less connectedness. But either way, some of these nodes in this particular little example would tend to be particularly important. So we started working with these kinds of problems a while back, looking at the epidemic networks. So here's an example project with Peg Margosian, where we're looking at the connectivity of the American agricultural landscape, where here nodes represent counties, and then the links between the counties represent a measure of the probability of movement between them by an organism that's species specific in this case. And so it's just based on how much of the host is available. So for example, so here for maize, the red areas here are the areas that would be connected for pathogens that require at least a low maize density to spread. And then if you look at different thresholds, and then up to here where red indicates the areas that are connected for pathogens that require high maize density to spread. So you can think of this as a risk factor, right? These are high risk areas if you got some kind of new introduction. And of course, then it's interesting to think about. So this was for the conditions in the 7 years ago. But so then, of course, it's interesting to think as maize production might move north to some extent, how do these structures change? And of course, that was very general. So drawing on information about a particular disease, here we're working with soybean rust data. So and this is work with Scott Izard, and people have already made reference to the IPM pipe data. So this was a project where we made use of that, where lots and lots of different cooperators, extension agents, and researchers collected information about soybean rust throughout this area over many years. And so then we were able to use that in modeling projects. And so part of the point here was that we were modeling the network in the United States for movement of soybean rust, since it invades again each year from the south, and trying to identify the points that would be most important for sampling. And we did find in computational experiments that if we use the network structure as a guide, that we can identify points that are more important for sampling if you're cutting back on the number of samples that can be included. And we also use this data set to look at epidemic evolution, as we were calling it, how this epidemic would change over time. Because of course, across the season, which nodes are particularly important is changing, but then also across years, we could see a difference in the structure of epidemics. And of course, this was over a relatively short time frame, so that we weren't really supposing that we were detecting climate change effects. It was more just the idea of maybe consolidation in the overwintering areas or something like that. But this approach could be interesting in longer term data sets as well, when you have such a luxurious type data set like this to work with. And then finally, another example for stored grain networks. So here you've got, if you're working at a smaller scale, you've got pretty defined nodes here, these storage structures. And then you've got different insect pests and pathogens and mycotoxins moving through the system as well. And then you have the risk of new pests being introduced, and then also the risk of pesticide-resistant insect pests moving through the system. There's been a lot of pesticide resistance developed in multiple stored grain pests. And here we had a nice data set available to look at wheat rail movement in the US. So 70-some% of wheat gets moved by rail in the US. So this is describing a lot of what's happening in terms of movement. So here the nodes are states, and the larger they are, the higher the wheat production. And then the width of the links between them is indicating the volume of movement. So once we have this structure together, then we can start evaluating nodes in terms of their inherent importance to the system. So if you know there's some kind of invasion, well, so you can think about trace back, trace forward sort of systems. Of course, if you find something at one location, you can think about how to trace back and trace forward. But also before you have a problem, you can think about what the key locations are to sample. Like in the United States, the central area is particularly important because it can go either direction. So there's a lot of wheat moving through there, and it can go in either direction. So by understanding the structure, you can identify some of the features like that. So here we had data and started out looking at the grain movement network. And then our interest is hooking that in with other elements of the system too, which we're just in the process of doing, like the management communication network. So part of that risk of the pesticide-resistant insects moving through there is poor management decisions. So if people are influenced to be more careful about selecting for pesticide resistance versus not worrying about it, that can be influenced by their position in this management communication network as well. So the general idea with the impact network analysis platform then is you've got some kind of management technology, and a socioeconomic network where ideas and money are being exchanged that's then linked to the biophysical network, movement of plants, pathogens, pests, and of course, understanding how those are connected is an important research question. And then together, then those produce some outcome like yield, profit, or resilience. Of course, it's a data-hungry type of analysis. If you really want to characterize a system, you need to know a lot. But if you don't know a lot about a system, you can start by asking general questions. Like one thing I'll discuss more later is this idea: if you've got a process like climate change, if you have some idea about the system structure, and then you have some idea about, for example, how many people have to adopt a management technique for regional management to be successful, then you could ask a question like, how does that change when you have climate change, so that maybe the probability of establishment at any given moment goes up by a particular amount? You could start to ask more realistic questions if you know more about a system, like how do changes in mechanisms for interpersonal influence affect the outcomes? And then if you know a lot about a system, like we had good information for the stored wheat network, then you can ask more specific and precise questions like what particular communication or land nodes are key control points for transmission through the landscape. So depending on how much you know about a system, you might sort of work through these different types of questions when evaluating a system. And of course, the goal of setting up this platform for impact network analysis is to try to optimize how you can take lessons learned in one system to use in another system as well. So once you know something about the structure, you can look at the communication networks, making sure key people are in place and the messages are in a form that can move through a network readily, have good incentives and regulations so that when people are making the decisions based on the information available to them, they'll tend to make the decisions that will benefit regional management, understanding invasion methods, networks to understand sampling and mitigation, and then understanding how those are linked together as well. So here's just an illustration for like a climate change example. So suppose on this axis we've got weather conduciveness to invasion. You might think about this as at each node in this network. And on this axis you've got the impact network component value that results in 5% chance of invasion. So that component might be something like the probability a land manager adopts a new pest management strategy, given that they're aware of it. So then you could see what this is like, and then if you know that the weather conduciveness to invasion is moving in this direction, then you could have in mind, maybe there's a threshold or some other factor you need to keep in mind about how many people are going to have to adopt a management strategy to compensate for the greater conduciveness. And of course, from a standpoint of resilience, the lag time in responding to important weather events help to determine how big the response needs to be to return to at least some acceptable level of management. If we think of our current level of management as acceptable, of course, that's a whole other question. Just to give an example, Xanthomonas wilta banana is an increasing problem in East Africa, Central Africa. And one big issue of it is that there's not currently resistance in most banana strains, so it makes it really important to keep it from spreading to farms, and it makes on-farm management really important. So it's spread by insects, contaminated farm tools, and infected planting material. So there are lots of different strategies for managing it. Some are too labor intensive and farmers aren't really interested in using them. But one technique people have been using pretty successful is called single disease stem removal. Where instead of removing the whole banana mat, which is really labor intensive, people just remove the stem that's infected, and that seems to work almost as well as removing the whole mat, and people are more likely to really do it. So if you think about that in the impact network analysis framework, so there's a disease management component like single disease stem removal, and it works pretty well, but people have to understand how to apply it because you can apply it badly. If you cut off the stem high, then it can actually be worse than not doing anything because the bacterial ooze is available to spread around. The communication network where people learn about it, and the decision network where in some regions there are actually policies that are forcing people to use it, and other areas people might not be aware of it at all. And then the dispersal network influenced by it and the establishment network. So here the different layers include the communication networks, the insects that spread it, birds, bats, small ruminants can spread it as well. and then farm workers that may be spreading inoculum on unsterilized tools. So here, in an analysis of that system, more on the realistic end rather than the precise end, suppose this axis is the probability that single disease stem removal is successful and applied, and we're just assuming that tools are sterilized with a probability at 0.7. Then over here, there's a question of the minimum probability of adoption of single disease stem removal such that disease incidence stays below 5% the majority of the time. So, so how many people have to adapt it for it to be successful, given that even if people adopt it, they won't be necessarily successful applying it? And so here we can differentiate between what's happening in the lowlands and what's happening in the highlands. So in the lowlands where there's more insect spread, there's a higher requirement for more people getting on board with using the technique. While in the highlands, it's easier to get by with not everybody adapting it, or not everybody, adopting it, or not everybody being successful. And these you could take as, to some extent, climate scenarios as well. The highlands will probably tend to become more like the lowlands over time. And I'll just mention for a second, because we often assume that resistance genes will be available spreading around, but it seems to me like that's not evaluated very formally, very often. So we've been working on this some, just characterizing global crop breeding networks. So for cassava, for example, potato, rice, and wheat. And so I'm just throwing those up there. There are a lot of implications from the structure. Well, and of course, things like there are no private cassava breeders. But then, of course, for other crops like maize, private businesses dominate. So that might be an interesting topic for more discussion too. But just to conclude about the impact network analysis part, so this idea of working through the different types of questions depending on how much data you have, we're applying this to other systems like laurel wilt of avocado seed system implementation. And it would be very interesting to us to think about other systems as well. And we're developing an R package to perform impact network analysis that should be available soon to make it easier to look at these integrated networks. And then finally, just to bring up the CCAF CGIR scoping study. So CCAF, Climate Change, Agriculture, and Food Security, is a program within the CGIR. So if you're not familiar with CGIR, it's a global system of international research centers. Like maybe the most famous ones are the Green Revolution centers that produced wheat and rice. Well, so interesting talking about adaptation. They were adapted so that they would have sturdy stems, so that if you applied more fertilizer, they would still stand up and not lodge. But they do a lot besides just sturdy stems as well, of course. So this was a team of us looking at this and coming up with some kind of a recommendation. Maybe this is in some ways parallel to what we're doing, coming up with a recommendation for what a group like CGIR, which has as its mandate to help resource poor farmers, and in this particular case, adapting to climate change. So what can we do, or what would we recommend that they do? So I'll put this in pieces, but basically we're talking about some different levels of types of activities and what we'd hope to accomplish. So first, just looking at weather and climate data as input information at different scales, where, of course, for some pest and disease models, especially ones that were designed for decision-making within a season, within a field, tend to have very high resolution requirements in some cases. And so are there ways to adapt those where you don't have such high resolution weather data necessarily? And then what can communities like, well, probably like our community here, as well as the community in CGIR that works with modeling and climate change adaptation, what can we do? And so this idea that often what's missing is a generic disease or pest platform to interface with crop models and weather data. And often access to the weather or climate data is not necessarily easy either. So this idea of developing a platform to make it easier for a person who's knowledgeable of a particular pest or disease to link into some system where they don't have to become a climate expert themselves, and having some specific models for the disease or pest that are iteratively improved, and then also ones that are specific for geographic locations that are iteratively improved. So this is all coming back to the idea that we're adapting to climate change, not just breeding better varieties necessarily. But even if there is no climate change, you might still want to do this kind of operation to understand what's happening in a large region where you can't necessarily sample regularly. And then once you understand this, what can you do to inform decision makers? So you could hope to influence farmers' chemical, cultural, and biological decisions, their choices about crop species and varieties, potentially inform insurance that can cover their financial risk, and inform donors and scientists and policymakers' prioritization. And then ultimately, so leading to the goal for all these programs, right? Improve food security, livelihood, and environment. Probably we need some more steps on here, right? For this to actually come to fruition. So if you all have comments on that, that would be very interesting, and I could incorporate ideas. And maybe this is interesting also for our discussion here for tomorrow. Like, what do we think should happen? Like I was saying, this is all focused on adaptation as opposed to not necessarily the no regrets perspective. Like from the no regrets perspective, you could just say, well, we need more and better, perhaps, crop breeders, et cetera. So this is really focused on adaptation per se. So that would be great if you all have any comments on that. And I thank you for your attention.
Thanks, Karen. That was pretty extensive. And I just had a question because of our, and I talked about our center, you know, we are definitely looking at things related to, and I'm, logic model kind of information. And I see a lot of that in here when you talk about the, the impact network analysis. And one of the things that we've always we're working on, try to understand is how much information is needed before knowledge is gained to then make the behavior change that we're interested in a farmer or grower or someone doing. I mean, is that known? Are you working on that aspect, or where are we with that?
Oh, let me see if I understand your question. Then, so, like, at what point do you know enough that you would roll out a product for a farmer? Is that the question, or?
Well, so at what point does the farmer make the decision that you want them to make? So a change in practice. So say a farmer is not doing something that is, environmentally or economically viable. What amount of information do they need to then change their practice? And so IPM. So we're trying to figure out how much they need to know to adopt an IPM practice or framework. So, and I don't know if that's something that you've worked on quantifying or trying to figure that out. I mean, okay. Does that help at all?
Yeah, that's interesting. Well, so because I guess there's some process of the scientists knowing enough that they feel confident that it's worth trying to convince the farmer to do it, right? Which is like the first part, I guess. But then how much does what would actually stimulate the farmer to make that decision to adopt it, assuming that it's really convincing to us it is good for the farmer?
Right.
Yeah, so that's a really interesting, social science question, right?
that's what a lot of your talk was kind of spurred that. So I don't know where we are with that, or if that's something you've been working on, or.
Yeah, so so I've worked as an epidemiologist for a while, and I'm trying to become more of a social scientist to look at those links, but I don't know if I could reply to that very definitively. It seems like it would be pretty situation specific, like in maybe a resource poor farmer scenario versus a wealthier farmer in the U.S. Could be a pretty different scenario. But it does seem like it's often the case. There are those interesting studies of like early adopters, late adopters, and so on, those distributions. And it does seem like it's often the case that there can be sort of a critical mass of people adopting a technique and then other people decide to use it. So I guess that's one strategy, is to come up with convincing cases, enough convincing cases that people are convinced to join in and also use the strategy.
Right, because we say, well, what is the impact of this program? What is the importance of this program to continue on? And so to quantitatively assess that, we need to say, well, because we did this, we saw this change in behavior. But it's hard to relate that change to that one program. There's so many things that are influencing decisions that you can't say that, yet we're forced to do that because we need to say why this needs to continue. So I don't need to go on that. If there's other questions, I just wanted to bring that up. So
maybe adding on to that, I mean, I think one of the challenges that we face right now is there's an incredible amount of information available, but it's highly decentralized relative to what it was several generations ago. And again, this is really with respect to how do farmers implement new practices. So it used to be they went to the extension agent kind of had the knowledge and it was disseminated. Now it's all over the place and there's multiple actors sometimes saying different things. So how do you take that into account in these networks and say, how do we really truly drive what needs to be done when people are getting information from an incredible array of sources?
Yeah, that's a very interesting question. I can solve it for you. Oh, well, not really, but I can comment on it. Yeah, that's it. Well, maybe sort of tangentially related to that, but maybe also sort of key is that people have made reference earlier how often there are lots of different sources of information that aren't integrated. And I've been a part of a lot of discussions, and I think it's a good point that, for example, if you were developing big genomic databases, you would, in order to publish the results, you'd have to put them in a data repository that is at least hypothetically something that anybody could access. But maybe it's partly because those technologies evolved at the same time as the genomic technologies evolved at the same time as the IT technologies that would allow people to do that. Well, people have been observing weather and pest or disease relationships for a long time, and we're just not in the habit of making these centralized databases. But if there were some way, I mean, that could be an important policy impact, would be if there were some way to make it so there were really good incentives for people to put their data in repositories where it would be easy to access with good metadata and so on. That could be a good policy outcome. But people have talked about that before. I'm not sure how to do it. Journals could require it, but then all the journals would have to agree that they'll require it. So.
Any other questions for Karen? Yeah, Keith.
This is a little different kind of question. So last time I heard a presentation on impact network analysis, though it was in a different field. The person ended their talk, which was really nice, by saying, and there's this one assumption that was kind of built into this that I'm really looking forward to go back and address and then take my network analysis to the next level. Do you have one of those kind of assumptions for this, or do you feel that what you presented for that network analysis was kind of mature and inclusive, or is there a next piece to add to the puzzle?
Well, I think that for any system, there's a question of when you would get diminishing returns from knowing more. Well, that idea of what's the optimal level of complexity to include in your model, which is sometimes you have to pass that to be able to go back to. Because if you have too little complexity in your model, you don't necessarily know that, right? If you go far enough that you realize you have too much complexity, more complexity than you need, then you could go backwards. So I guess that's related to your question, right? How do you figure out when you've included enough factors that your result is realistic? And of course, if you keep scaling up higher and higher to broader and broader spatial extents, you can keep incorporating more and more factors, and then you can have a new hybrid arise that has a completely different host range, right? So I guess there are always factors like that, and it would depend on the question, and maybe you have to frame the question kind of precisely, like assuming new hybrid varieties of the pathogen don't arise, how do we think this system would work?
Okay, good. Oh, Matt, you've got a question?
Yeah. I think that it's not necessarily mutually exclusive, that this is you're advocating what is also a no regrets option. Disease is about epidemiology. And it may not be this disease or this insect, but whatever it is, it's logical that interrupting the flow and controlling things when they're rare is in the best interest of a broader community. And so I think you can develop that in a way that it might make sense. I don't know if are you familiar with the viticulture communities in Europe, like in Spanish denominaciones, the name on the bottle. Those are self-organized units that they're actually all about pest control and other things, but a lot of it is about pest control. They're older than any government in the world. They've been operating for hundreds and hundreds of years because everyone views it as in their own best interest. And maybe having some, looking for parallels with something that evolved from the bottom up like that and has been self-sustaining might provide some inspiration for organizing the thinking and presentation of these networks.
Yeah, that's interesting. The self-generating networks are often sturdier than imposed network structures, right? Well, and as you were saying, it was making me think too about this idea of the no regrets approaches, because one issue is just that's pretty broad is when talking to a colleague who was setting up a global network to evaluate one particular pathogen, and then was saying that in a lot of countries it was hard to find the person who would be part of this network, and that one country had no one with training in plant pathology. So you could argue that a no regrets approach would be to just have someone that is knowledgeable of plant pathology, so when a new pathogen arises, they will have some idea about how to deal with it.
Okay. Walt, you got a comment?
Yeah, this idea of modeling for no regrets, and at least my experience in dealing when working with growers is not that there's a no regret, it's that they're trying to minimize the maximum regret. That they have certain resources that they're willing to risk and certain resources that they're not willing to risk. And they're trying to walk that line.
Yeah, that's well, and coming back to the idea of what scale you're thinking of, right? Like a grower in a season or in a small number of seasons versus globally, I guess the definition of regret would change, potential regret would change a lot across those scales.
Yeah, good discussion. We're going to have to
Thanks.
end and move on to our next speaker, who is Steve Frank from the North Carolina State University, and is going to talk about insects and some of the work he's been doing in that area. So Steve is behind me.
I am sneaking up.
Sneaky guy.
All right. Thanks, folks, for hanging in there. Hope everybody has a little coffee, and I'll try and be as perky as I can. I'm not known for my perkiness, but we'll do this. So I'm going to talk about some work we've been doing for the last several years, looking at the effects of temperature generated by the urban heat island effect on pest insects and on tree health, and the extent to which we can use this to predict the consequences of global climate change more generally. Start with this picture, and I like old stuff, and these old scientists, and this gentleman name is Joseph Putnam, and he lived about 100 years ago. He died very young of tuberculosis, but the reason that I start with him is that he came up with one of the first examples that I can find that shows the relationship between urbanization and native pest outbreaks. And so what Joseph Putnam described in 1890 or so is that this native cottony maple scale was super abundant on maples in downtown Davenport, Iowa. And when he would go jaunting through the countryside, as gentlemen of old had time to do, he would never find this scale insect on the maples in natural areas. And so, here's Davenport, Iowa in around 1900. Not exactly a thriving metropolis, but you can see the way the trees are planted there, the same way that we plant them now. And since then, in the last 100 years or so, lots of folks have made this observation that were reviewed in 2010 or so, that a lot of insect pests become more abundant on urban trees or in urban landscapes than on the same plants in natural areas. So our hypothesis has been, although folks have approached this from many directions, our hypothesis has been that temperature could be one of the driving factors of this phenomenon. And so whether you're in Davenport, Iowa 100 years ago or Times Square today, that the thermal differences between rural areas and urban areas could be responsible for these pest outbreaks. And so the pests that we're interested in are these native chronic herbivores. Okay, so these are things that exist in low levels. You can find them in the woods, you can find them in natural areas, but you've got to look really hard oftentimes. These aren't exotic outbreak type of pests that we're interested in. And we focus primarily on scale insects. And so this is one of the scales that we work on. This is called gloomy scale, and this is a southeastern, it's distributed in the southeastern U.S. Historically, and it's a pest of red maples and some other trees, but primarily on red maples. It's got one generation per year. This is my student Adam Dale. He's now at University of Florida. I guess I can look over there. I don't have to look. So this is the Oaklicanium scale, another native. This is a soft scale. The gloomy scale is a armored scale. Pest of oak trees, primarily willow oak trees, which are with red maples, two of the most commonly planted street trees. Again, native to the U.S., univoltine, and for my presentation, not being a modeler, this is the most you'll get of this is upscaling, this is downscaling. Right? Just to clarify some jargon. All right. So the way that we've approached this is we take these surface temperature maps that we get from NOAA. This is Raleigh, North Carolina. Just to get you situated here, this big red blob is the Raleigh Airport, okay, which is about 10 degrees C, warmer than this state forest right next door. And so we take these surface temperature maps, and you can see even within downtown, there's a lot of heterogeneity in temperature. We overlay tree inventories that we get from different cities. And so this is a map of all the willow oaks in Raleigh. There's a lot of them. But what we can do then is compare willow oaks in these hot spots to willow oaks that are just a few blocks away but live in a different thermal environment, often by several degrees. Okay, the punchline of all this is that, here's our gloomy scale. Gets 200 times more abundant with a temperature change of about 2.5 degrees. Okay, so almost no gloomy scales down here. This is mean 7-month temperature recorded in the canopy of the tree. Almost no gloomy scales at this end. Several hundred gloomy scales per inch essentially up at the other end. Okay. Same thing with our soft scale. The way that Emily Meineke is a graduate student who did most of this work. She's a postdoc now at Harvard, and she divided her trees into hot trees and cold trees, essentially. And again, whatever life stage of this critter we look at, you've got 10 to 12 times more scales on trees that are a couple of degrees hotter than the cooler trees. So one of the things, a lot of what we've talked about in this session, it seems every talk at some point comes around to invasive species, and so I've started to think about whether these critters are invasive, right? And they have some of the characteristics that, you know, different people have different characteristics of invasive species, and I'd love to hear, you know, I'm not a invasive species expert, but, you know, having no evolutionary history with an ecosystem, almost no living thing has an evolutionary history with a city, right? It's just, it just doesn't. Maybe some microbes, maybe some cockroaches. There are things that maybe have evolved within the context of city, but generally no, right? This didn't exist. High reproductive rate, our scale insects have this. Our soft scales produce 2,000 or 3,000 eggs. They can produce sexually or asexually. High dispersal, when these crawlers come out, they can blow around the city on wind currents or just rain down on the plants below. Okay, species richness is low when these are present, and they have negative effects on human interests in terms of destroying shade trees and the aesthetics and the economics that go with that. So a couple of other commonly cited invasive species characteristics are that they've got some phenotypic plasticity that helps them adapt to a new environment, being a city in this case, and that there's some disconnect between the critter and the local natural enemies in that habitat. So our hypothesis for this section is that native scale insects do become invasive as defined by this set of characteristics due to this warming effect. Oh, here comes all this stuff again. Uh-oh. Okay. So a couple of, just an outline of how I'll go through this. I'll address the question I just brought up about whether these species become invasive due to phenotypic changes and disconnect with natural enemies. Talk about the work we've done to see if cities can predict climate change. And then, you know, we're interested in trees. We care about these pests because we care about trees and their effect on tree health and the services that trees provide. So gloomy scales, you can see how abundant they get on these trees. Each of these bumps is a scale insect. They live under a waxy cover. And what Adam did one day was started opening these things up. They're about 1 millimeter across, opening them up and squeezing out their embryos. Okay, so that's what these little blobs are in here. And so in terms of phenotypes, Adam found that when he would measure these critters before he squeezed out their embryos, the body size of the scale insects increased along the same 2.5 degree, I can point anywhere that's easier for people, along the, I can't win with the pointer. Along the same 2.5 degrees of warming, body size increased by about 1/3, from around 1 millimeter to about 1.25, 1.3 millimeters. So we've got this phenotypic change. And again, here's a scale insect with its cover removed. Here's the embryos again. And here's the relationship between, again, the same temperature on the x-axis and the egg count inside of those critters. Okay? And so again, you go from around 10 eggs per female on the coldest trees to, 30 or so on the warmest trees. So we've got this that helps explain some of that, the great densities on the urban trees. This is the oak lecanium scale that Emily works with, and what she did, remember, she found, much higher scale densities on her hot trees, and so to try and sort out whether this was, just an effect of temperature on insects in a general sense, or whether there was some sort of plasticity or adaptation going on. Emily collected these egg sacs. It's not a true egg sac, it's a female with the lays anyways. It's full of eggs, okay? And she collected these from hot and cool trees, and then she conducted an experiment in our phytotron facility where she grew trees in, she put eggs from cold trees in cool growth chambers, eggs from cool trees in hot growth chambers, eggs from hot trees in cool chambers, eggs from hot trees in hot chambers. So she has a full factorial of hot and cool chambers and egg origin. And what she found was that here's the cool house, the cool chamber, and the hot chamber. And these are scales after it was around 9 months of growth on the trees in these chambers. And so hot scales in the hot house do extraordinarily well, okay? But cold scales from cold trees originally, gain really no benefit from being in a warmer environment. And likewise, scales from hot trees they don't do that much better in a cold house. And so there's something going on with these critters, and we don't know if this is, just a maternal effect, some sort of plastic response like that, or whether, since we've got sedentary critters that can reproduce asexually, whether there's actually some adaptation going on in response to the thermal environment that they find themselves. But again, a difference then in the phenotype of survival, essentially. Okay, so natural enemies, we're going to look at some of these parasitoids on Emily's soft scales here. And the life cycle of this scale insect, they spend, well, we'll start here. Here's these egg cases. Underneath it that these are produced in April and May. There's 2,000 or 3,000 eggs inside of there. When those hatch, little tiny crawlers go out and they spend the summer on the bottom of leaves. Before leaf fall, they move back to the branches to spend the winter. And that's their yearly deal. When the females are producing eggs, in this time, there's a whole slew of parasitoids that can attack them and reduce egg production, essentially. And so Emily focused on this life stage to study the interaction of these parasitoids with this scale. Okay, here's some just beautiful parasitoids. And so Emily went out weekly. On this axis, this is time, okay, every week from March 5th over to April 16th. And in this figure, you have the proportion of scales on her red hot trees or blue cool trees that are producing eggs. Okay, so this is phenology. At this point in early March, nobody's producing eggs. As you move this direction, these hot trees start producing eggs. Many more scales are producing eggs than on the cool trees. So there's a difference in the phenology of the scales and the time of year that they produce eggs. When you look at the parasitoid abundance in these trees, there's really no difference in the phenology of the parasitoids. Okay, so you don't have this sharp spike in that would indicate earlier or faster parasitoid development or emergence. So you've got faster or earlier developing scales, but the parasitoid community is not, does not do that. And so in this figure, the other interesting thing is that we had looked at for a couple years before we figured out this interaction was that the proportion of parasitism is equal on hot and cold trees. And so for several years, we would collect scales, rear out the parasitoids. The percent parasitism of those scales on hot and cold trees is the same. It's always the same. And so that's what this is, proportion of scales with parasitoid larvae, okay, the same for your red and blue lines. The difference then comes in that if you compare scales that are not parasitized and the number of eggs they've produced, they produce almost the same number of eggs, no significant difference in the number of eggs that hot and cold scales produce. But when you look at scales that have been parasitized, this is where you see the difference, with parasitized cool scales producing fewer eggs than parasitized hot scales. And part of this is because those hot scales, by the time the parasitoids come out, may have produced 1,000 or 2,000 eggs already. And then they get parasitized, but the female's work is done at that point. So there's this phenological, heat-driven phenological mismatch between this critter and the parasitoids in its environment. Okay, so we got, working on this for a couple of years, we got interested in whether the response of herbivores, we're talking about, in all these cases, 2.5 degrees of warming, which is the amount of warming predicted over the next 50 or 100 years. And how well does this response mirror what we may see in natural forests? So my research associate, Elsa, who's phenomenal, she disappeared for a couple of weeks one time, and I didn't really notice. That's just the way I am. But she came back, and she had been all over the southeastern United States visiting herbaria. Okay, so this was super cool. And what Elsa realized was that you could take herbarium specimens that are even 100 years old, and because we're dealing with these armored scales that have this non-living waxy cover, they're still on there. And so we can find the scale covers just like you may find a turtle shell in the woods or something, and we know that those scales were there. So here's another picture of these herbarium specimens. Here's the scales that you can see on there. Here's one of our urban trees and the scales on there. And the great thing about botanists, I don't know why they keep collecting red maple branches. If there's a botanist in here, we could talk about that. But they do. They do it all the time. And lucky for us, I mean, I guess entomologists still collect bugs, right? But we know the red maples at this point. Anyways, we've got a slew of these. So here's all the sites throughout the southeast from where we have herbarium specimens, because the botanists, dutifully mark down the crossroads by Jim's farm or whatever where they found these things. So we have all these sites, and just looking at, in this figure, on this axis is gloomy scales per inch on urban trees in Raleigh. In this figure, gloomy scales per inch on herbarium sheets. Notice the scale, the magnitude is different of those, right? But the shape of the response that she found was very similar, okay? And so here's the black dots, and I don't know how well you can see the black and the blue in this figure, but they follow the same trajectory up. So Elsa went back and looked at this another way. The great thing about Elsa is that she's very, well, there's lots of great things about Elsa, but she's always convinced that it's a fluke, and I'm like, Elsa, that's great, write it up. She's like, no, it can't be. It couldn't possibly be, right? So she looked at this another way, and she took running, I think these are decadal average temperatures, okay, and then she looked in each of these hot periods and cool periods, so hot decades and cool periods. And these diamonds are just the proportion of her herbarium sheets that were infested from those hot or warm decades. Okay, and so in this first warm decade in the 30s and 40s, I've got to look over here and point over here. About 30% of her specimens were infested with scales, so this is just presence, absence of scales. Then you go down to about 20% in the next decade. Warm again, up near 40% of her specimens infested, and back down to 20 in the next cool decade. Okay, then of course we've got this figure that we've seen in every presentation for the last 2 days, right? Where all of a sudden you've got this rapid, extraordinary increase. So what happened there? Well, Elsa disappeared again, and when she came back, it turns out she had gone and tracked down 30 of these, well, 20 of these sites that she had detailed enough information and that were still natural forests that hadn't been developed, which in North Carolina happens pretty quickly. And so what she found was, I apologize for this axis is messed up, but each of these lines is connected by 2 dots, which is the left-hand dot on each line is the herbarium specimen, scale abundance per inch, followed by the new collection that Elsa did from the same spot. Okay, and she would, again, this is Elsa, she'd collect them, she pressed them on herbarium boards, she put tape so there would be the same amount of area, she did the whole thing, right? And what she found was that in 16 of these cases, the black lines, scale abundance went up from when it was collected. In these yellow 2 cases, scale abundance stayed essentially the same, and in 2 cases she had less scale abundance. But the overwhelming response was that visiting the same natural forests in the same spots, she found more scales decades later, presumably warmer climates. Well, we know warmer, we know the temperature of all these. Okay, so we're working more on this. Anybody who's jazzed about this idea, or not, if you think this is the craziest thing, tell me that too. But you know, this is one of the directions that we're going. Because you've got cities everywhere that could be experimental units. It's hard to replicate warming experiments. They're extraordinarily expensive, and for that expense, you tend to get a pretty small experiment, right? If we can use cities that have been warmer for hundreds, maybe thousands of years, we may be able to make more headway with certain critters where it's appropriate. Okay, so how do pests and warming affect tree health and services? So looking at our red maples with gloomy scales, you know, we've got maples in all different kinds of conditions here. Here's examples of, urban foresters have this rating technique. They can go out and rate trees very quickly as poor, fair, good, or excellent. And so all the tree inventories that we use have these ratings associated with them. So we've got, you know, the geographic location of the tree, we know the DBH, we know the condition, and often a bunch of other stuff about those trees. So just as an example, this is what an excellent tree looks like. Okay, here's a poor tree. It's pretty clear the difference. Sparse canopy, dead branches. It's just generally messed up. So Adam, he had these study trees all over Raleigh that he knew the condition of, and he knew the scale abundance, and he knew the temperature from his data loggers. And then he went back and he used the tree inventory to investigate all the trees, of all the red maples of Raleigh. where again, we know the DBH, we know the condition, and from the Landsat, we know a relative temperature that those trees exist in. Okay, with Adam's study trees, this is kind of a wacky figure, but essentially as you move this direction with scale abundance, you get a probability that a tree is in one of these conditions. And so as you move, to higher scale abundance, the probability of finding a poor tree, that a tree is in poor condition or fair condition, are really your only two options. The probability of finding an excellent tree or a good tree diminishes to not very much. Okay, likewise, if we look at, these are all the red maple street trees in Raleigh, and pull out some of those, we get a similar response where excellent trees, just sort of simplify his results, excellent trees tend to be in cold locations. Poor trees are much more likely to be in hot locations. Oh, there you go. So with Emily, she took this a little bit further, and she's got a paper coming out this week where on her willow oaks she measured a whole slew of physiological functions and photosynthetic rate and stomatal conductance and these kinds of things on her trees. And again, just to get to the meat of this, as temperature increases, again, we've got about a 2.5 degree span of her trees. Photosynthesis declines, and basal area growth also declines along that axis. So Emily was interested in, there's some formulas out there, like with this iTree program and stuff, where you can punch in a tree DBH and get all kinds of values on how much carbon it sequesters and how much water it collects and this sort of thing. So Emily dug into some of the allometric equations that they use to create those, and incorporated the effect of temperature that she found on, as it affects photosynthesis, basal area growth, and of course, insect abundance. And so we used, again, our database of willow oaks from the city of Raleigh, surface temperature maps, all kinds of allometric equations and magic. And then, the essence here is that on her hotter trees, the warming, if you take all those trees in Raleigh, warming reduces the carbon sequestration of those hottest trees by about 12%, which is about, just for willow oaks on streets in Raleigh, is about 27 metric tons that are lost, which isn't a lot in a global sense. I know some of this corner of the room, well, that's nothing, right? And it's not a lot. I mean, it seems like a lot. It's a lot. If that was just like coal in a bag, that would be a big bag, right? But again, this is just street trees, one species of street tree in one city that we're working with. Okay, so we all know by now climate change can change insect physiology, behavior, and biotic interactions. We're working to see how well we can use cities as sentinels to predict range expansion, to predict which chronic pests may become important pests in the future. With gloomy scale, we find it way above where the original range maps indicated it was. Up into Delaware, and we find it in Manhattan now, and other places where it didn't used to be. But in the cities, we find it in Asheville, where it didn't used to be, downtown Asheville, which is up in the mountains, but you don't find it in the forests around Asheville yet. So, Hicke heat and pests combine to affect carbon sequestration, and I'm interested in how we may be able to build this into Earth system models or something, something fancier than what we do. So thanks for listening. I know it's been a long day. I'll take any questions.
Okay, we've got a few questions from, we got one question from online. So Nancy, you want to read that?
And it's going back to the beginning of what you were talking about with the trees. Given those differences in scale growth between hot and cold trees and hot and cold places, could there be difference in the tree's physiology, like bark thickness, sugar content, fluid flow rates, themselves growing in the two different environments that support more or less scale growth and reproduction? Could it be the host rather than the pest that is different?
Yes. Next. No. So all those things are true, right? When you've got higher temperatures, we've heard about the evaporative changes. We know the stomata close up earlier in the day on those hot trees. And you can have, when we measure the vascular pressure and things, they're under more drought stress typically. And so there are physiological differences in the tree. We've accounted for that in some experiments using, we've got a set of trees that we've watered for 2 years. And you see the response of scale insects in terms of fecundity. The response to temperature stays the same in slope, but the intercept on watered trees is lower. So there's a combined effect there that we're still sorting out.
All right.
Steve, just one follow-up. I'm John Garrett. So what's the main mechanism of defense from plant perspective?
So we honestly don't know. The main mechanism of defense against these scale insects, I've got a postdoc now who's working on some of this, and we don't see a big change in the nutrition of the leaves. We've got to get in and measure the nutrition. The armored scales aren't feeding on the leaves, they're feeding in parenchyma cells. So the exact mechanism of defense is unknown at this point. But it could be less if they're dedicating more carbon to other things.
Lou, you got a question?
Yeah, thanks. That was a really fascinating presentation. I enjoyed it very much.
Thank you.
And I'll put in a plug for using cities as sort of a surrogate for climate change, because we've done a little bit of that as well. Taking the same seed bank and putting it out in downtown Baltimore and then in the suburbs and then in the country, and seeing very clear phenological changes in terms of plant development and a much greater increase in invasives in the seed bank becoming to fruition in the city. One of the questions we had in that, and I'm curious about this, is that cities, in addition to being heat sinks, can have sort of a dome of CO2 associated with them as well. And I know for insects that CO2 can be an attractant factor. It can act to bring insects in. And one of the things we found in cities is that the CO2 there's a lot of CO2 variation. Sometimes it can be quite high and sometimes it can be very close to ambient. Is it possible?
In space or time?
Actually, in terms of time, we tend to find that, well, in both actually. Spatially, you can see some areas that are very high, but only for short periods. And the question becomes, is that, or could that be an attractive, sufficiently high enough for it to be an attractive factor for various insects to bring them in? Do you have any kind of sense of that?
So I don't have a sense for how, well, okay, so with our organisms, these scale insects, they don't move very, they don't fly. They wouldn't be migrating from the outer areas in response to a CO2 gradient. And so they're pretty sedentary. They sit there, they produce their eggs. The crawlers either crawl out or blow away. So they really don't have behavior to follow a gradient like that. Other critters, though, potentially that could be important. We've got Elsa has work now where she's taking air quality measurements. She's working on a project looking at nitrogen deposition and CO2 gradients on flower quality and pollinator attraction. So she's collaborating with somebody who's got a mobile air quality lab. They're going to go out and measure these things around Raleigh. So we'll get a better handle on that, what those differences are.
Other questions? Ben?
Yeah, what about air pollution? I know in a lot of these cities, and there's plant physiologists at Columbia who have been studying this, looking at like ozone, differences in ozone composition, in the suburbs versus versus these urban areas, and the effect that that can have on tree health.
Yeah, so there's certainly differences in pollution between cities and country, rural areas. The way we set up our experiment was that, all of our trees were in the city. And so we've got, our hot and cold trees could be a quarter of a mile apart. And so so they're not growing in vastly different environments the way that, they did at Columbia. And, and there's been other papers showing that tree growth should increase in cities. But those studies didn't include biotic effects of pests, and oftentimes trees are grown in containers or something like that. So there could be benefits in the absence of pests, and maybe this is where pest-free plants like exotic trees may do great in cities. Maybe that's what we should be planting.
Can I respond to that?
Yeah.
One of the things that when we did our rural-urban transect, is we were obviously very concerned about ozone being a factor in terms of affecting plant biology. And one of the things that we found is that when an ozone event did happen, typically it would happen within the city or on the edge of the city, and then it would spread, but it would spread very quickly, so that the ozone levels would rise within usually 8 to 10 hours. So by the end of the day, anything within the rural area, say within 20 to 30 miles of downtown Baltimore, was also experiencing the same ozone levels of what was experiencing downtown. So while there could be in a very short term some differences, typically what happens is that any ozone event is affecting more than just what's happening in the downtown area. It's affecting a larger segment around the urban area. The biggest change we saw was actually nitrogen deposition. There's a lot more nitrogen being deposited in city areas than there is out in the surrounding countryside.
Other question? Walt, use your microphone.
In urban environments, you have a lot of radiative heat that actually makes environments extremely different just within 100 feet because of how it reflects off buildings, streets. And there's also the fact that within the urban environment, you have a lot of trees that are essentially container pods, because in street islands, you have a small amount of soil with a 2-foot wall around it that goes all the way down because of the gravel and everything that the roots don't penetrate. So you're dealing with a lot more environmental effects than just temperature.
Sure.
How do you pull that out of this type of data set?
So a couple of ways we've tried to deal with that. We include impervious surface area. There's a strong relationship with impervious surface area, and actually for our maples, impervious surface has a higher R-square. It accounts for more of the variation than just temperature alone, because impervious surface affects soil compaction, water availability, and other things. So statistically, we can start to separate out some of these effects. The other way that we've tried to do it experimentally is, again, by doing these watering experiments where we have trees in parking lot islands, trees in tree lawns, trees in devil strips, where in a paired design, we have one that we water and one that we don't, right? And so we can start to separate out especially the drought effects imposed by heat and impervious surface from just the temperature effects. And Emily did an experiment as part of this recent paper where, we've got an issue where it's hard to separate out the effects of temperature and other things from pests, because as temperature goes up, pests go up, and you don't have hot, pest-free trees. And so we had, again, in a paired design with her willow oaks, trees that we treated constantly with horticultural oil to reduce the scale abundance, and that allowed us to have, a gradient of temperatures that wasn't linked to pest abundance, to try and separate those two effects on tree growth, for instance. So, yeah, there's a lot going on. Reviewers never fail to point that out. But it's, if one of our arguments, if you look at warming experiments in forests, at Duke Forest and at Harvard Forest, two of the largest forest warming experiments, and they were 5 meters, right? And that's great. Like, we've learned a lot from those experiments, but you don't have trees that have lived their whole life in those experiments. You don't have, animals can move in and out at will. And so to the extent that every warming experiment has issues, we're trying to sort out ours.
Great, thank you. We're going to end this session with the break. So we've got about 15 minutes, and then I would encourage everyone to reconvene, and especially those folks that are online. This is your chance. If you've heard something over the past day, 2 days, that you want to comment on or ask a question, that would be the time to do it, because from here on out, it's open discussion. So let's take a break and then come back at 3:20.